Imagine having a DNA test that is as easy as using litmus paper. Can you believe such research began over a decade ago? Hey there, welcome to Science is You. We're a brand new channel with a mission to make real scientific research papers in molecular biology and medicine enjoyable and accessible to everyone. But we need your help to get going. If you enjoy these videos, please subscribe to help this channel get off the ground. Thanks for watching. Paper has a lot of uses. Writing, art, packaging, filtration. There's also increasing interest in bioactive paper. Paper that can bind and detect biological substances like proteins, carbohydrates, and DNA. There are a lot of possible uses for these bioactive papers, including smart packaging, with built-in indicators, filtration, and medical diagnostics. A well-known example of bioactive paper in medicine is the at-home pregnancy stick test. Paper can be made bioactive in a number of ways, like printing or coating with chemicals. The ability to detect and examine DNA is a big interest in medicine and forensics. Methods of DNA detection like hybridization involve binding of the DNA to a glass surface, which can take hours and lots of chemicals and equipment. But could simple paper change all this? This research study, written back in 2012, presents a new method for equipping paper with DNA detection capabilities. Did you ever think a substance found in plant cell walls could be the key to developing a paper DNA sensor? The researchers here found that a molecule called xyloglucan found in plant cell walls can be used to help DNA stick to the surface of paper. Also, they found that by adding amino groups to the xyloglucan, they can improve its ability to bind to the fibers of the paper. This made it possible to take a strand of DNA with an amino group attached and bind it to the xyloglucan on the paper. But how do we know if the DNA is really on the paper or not? Well, it's all about finding the complementary strand. DNA is made up of two strands that connect using different sequences of molecules called base pairs. Think of it like dancing. Partners can separate during the dance, but always come back together because they complement each other. So, after binding our sample of DNA onto the paper using this special surface treatment, we can detect it using its matching strand or complementary strand. But how can we see such small sections of DNA? That's where a fluorescent tag comes in. By using DNA with a fluorescent tag, we can detect whether or not its complement is present in a sample of DNA bound to the paper by looking for fluorescence. To do this, the researchers created small pieces of DNA called primers that specifically target and attach to certain sequences of DNA. They used polymerase chain reaction, or PCR, to make copies of the primers that consisted of one strand of DNA attached to a magnetic bead, and the other strand with a fluorescent tag. They then raised the pH of the solution, which causes the double-stranded DNA to separate into single-stranded DNA. The single-stranded DNA with the fluorescent tag was then isolated and used to detect DNA that is on the paper. Let's see how this works in an example. The authors of this paper created an array of single-stranded DNA samples attached to the paper in a grid pattern. At the top was a negative control, which was liquid with no DNA, and a positive control, which was DNA known to bind to their fluorescently tagged DNA. These controls were crucial to ensure the accuracy of the test. If there's fluorescence in the negative control, we know our test isn't working properly because it's sensing something other than DNA. Conversely, if we don't see any fluorescence in the positive control, we know that our test isn't detecting DNA when it should be. Beneath the controls, they deposited samples of either dog or human DNA and applied the complementary fluorescently tagged DNA for each by allowing it to wick up the piece of paper, called capillary transport. The results were remarkable, with signal observed only in the area where the complementary DNA sample had been deposited, indicating that the paper-based test was indeed capable of distinguishing specific types of DNA. Of course, this technology still has a ways to go, but what do you think? Will nearly instant paper-based DNA tests ever be as common as litmus paper? What would that mean for the future of medicine, forensics, and diagnostics? Don't forget to subscribe to help this channel get going, and remember,